Hello everyone, welcome to Lab Science Tutorials. In our previous video, we discussed the identification of gram-positive organisms. In today's video, we will be discussing the identification of gram-negative organisms. We all know that gram-negative organisms appear pink in the gram stain, and most gram-negative bacteria are rod-shaped, especially gram-negative bacilli are rod-shaped. Then we also have gram-negative Coccobacilli, Diplococci and curved rods. In this video, we will see how to differentiate each of them using their specific characteristics. This will help you crack most questions, especially the case study questions in your exam. Like I said before, gram-negative bacteria appear pink in the gram stain. We have four different categories of gram-negative bacteria. The first one is Diplococci, then gram-negative bacilli, Coccobacilli and curved rods. These are the four different categories of gram-negative bacteria. First, let's discuss about Diplococci in detail. The shape of Diplococci means two spherical or round bacteria are joined together in a pair. That's how Diplococci look under the microscope. Among the Diplococci, the two most important organisms are Neisseria gonorrhea and Neisseria meningitidis. How do we differentiate them? First, both of these organisms are Diplococci and both are aerobic in nature. We differentiate Neisseria gonorrhea and Neisseria meningitidis using maltose fermentation. If maltose fermentation is positive, then it is Neisseria meningitidis. If the maltose fermentation is negative, then it could be Neisseria gonorrhea or Moraxella. That's all about Diplococci. Now let's move to Coccobacilli, the second group. Gram-negative Coccobacilli are the bacteria that are short or oval in shape. Examples include Haemophilus influenzae, Bordetella petrosus, Brucella and Acinetobacter. Suppose if you get a question in exam and it says that gram stain shows gram-negative Coccobacilli, then you have to think of these organisms immediately. Next, we are going to discuss about gram-negative curved rods, the third group. These are very important. The three main organisms are Campylobacter jejuni, Vibrio cholerae and Helicobacter. How do we differentiate them? All three are oxidase positive, but we use certain specific characteristics. In case of Campylobacter jejuni, these bacteria will grow at 42 degrees Celsius. And Vibrio cholerae can grow in alkaline media, and Helicobacter pylori is the organism which can produce urease. So gram-negative curved rod which produces urease, then the organism will be Helicobacter pylori. That's how we differentiate them. Now let's move to the most important group of gram-negative bacteria, the gram-negative bacilli. The gram-negative bacilli appear pink in the gram stain and rod-shaped organism. How do you differentiate gram-negative bacilli? There are many organisms, but we use certain biochemical tests to identify them. First, we do lactose fermentation. If the organism is showing lactose fermentation, then it could be E.coli, Klebsiella, Enterobacter, Citrobacter or Serratia. Among this, E.coli, Klebsiella and Enterobacter are fast lactose fermenters, while Citrobacter and Serratia ferment lactose slowly or late. Now, if the lactose fermentation is negative, we see no NLF colonies in MacConkey agar. How do we identify those organisms? The next step is oxidase. If oxidase is positive, then and NLF colonies on MacConkey agar, then it is most likely Pseudomonas or Burkholderia. If the oxidase is negative, then it could be Shigella, Yersinia, Salmonella or Proteus. Now, how do you differentiate them? For the further differentiation of this oxidase negative group, we use the H2S production or TSI agar. If H2S is produced, it is Salmonella or Proteus. If H2S production is absent or there is no H2S production, then it could be Shigella or Yersinia. So this is how you identify the gram-negative bacilli. Please go through the flowchart thoroughly and remember all the key points. These points will definitely help you crack most bacterial identification questions. Thank you so much. If you like this video, please do like, share and subscribe.